Thank you for the patience, everyone, and thank you for the introduction and for the organizers for the opportunity to present my work today. So this is some work for my master's thesis. I'm very excited to share with you all. So as we've heard just a few minutes ago and throughout the day, PAX6 is a highly conserved transcription factor. It's a master regulator that plays a very important role in establishing the optic fields, among other roles in ocular development during embryogenesis as well as later in adult life. And so aniridia is caused by a heterozygous loss of function of PAX6, leading to a dominant haploinsufficiency of the gene. So this means that patients in their cells carry one wild-type copy of the gene, which encodes a properly functioning PAX6 protein, and the cells carry one copy of the gene, which contains a mutation or a variant, which disrupts the production of a properly functioning protein. So here I've denoted the most commonly reported patient variant that causes aniridia, denoted C718C to T, located in exon 9. And it's a single base substitution that results in a premature stop codon. So as we know, there currently aren't any vision-saving therapies available to treat aniridia, and thus there is a therapeutic need to be met. This, along with the monogenic nature of the disease, as well as the progressive nature, creates it to be a candidate for gene therapy. So one exciting approach is to utilize the gene editing capabilities of the CRISPR-Cas9 system to correct the pathogenic variants that cause aniridia directly at the genomic level. So the traditional CRISPR-Cas9 system involves an RNA-guided nuclease, which is directed to your genomic region of interest. And after introducing a double-stranded break into the DNA, we can introduce a DNA template into the cells as well to target the homology-directed repair pathway in order to confer a precise change in the genomic sequence. Unfortunately, this system is not perfect, and the non-homologous end-joining pathway can also result, where we get the introduction of random insertions and deletions, which can further disrupt the gene. Fortunately, there's been a lot of development in the field in recent years, which has led to the invention of an adenine base editor. So this involves a Cas9 nickase, which has been engineered to be able to only introduce single base cuts into the DNA backbone. This is fused with an adenine deaminase, which can confer the precise single base conversion within a particular editing window, all without introducing double-stranded breaks into the DNA backbone. So as we think about how we aim to develop and optimize this technology for the purpose of treating humans, we have to think about how we can translate this work from the in vitro proof of principle stage to a small animal model, to non-human primate studies, and eventually patients. And so from the initial proof of principle stage, we have to ensure that we are able to translate the therapy to human DNA. And so the strategy that the Simpson lab has taken is to minimally humanize our region of PAX6. And we do so by utilizing the CRISPR HDR system and delivering these reagents to mouse embryonic stem cells to confer the exchange of the mouse-specific sequence with the human-specific DNA sequence here. And so we pick individual clones of these cells in order to establish a genetically homogeneous cell line. And so through this work, we're able to characterize these clones by PCR assays as well as RFLP screens to derive 3 novel cell lines, each of which are homozygous for the humanization. The first cell line is homozygous for the patient variant, the second is heterozygous for the patient variant, and the third is homozygous for the non-mutant sequence to serve as a control during therapy development. So thinking along in the same idea about translating this work towards that pipeline from bench to bedside, we have to ensure that this minimal humanization event doesn't disrupt the function of PAX6. And so the lab successfully established a humanized non-mutant mouse, or referred to it as He9, and I characterized this strain to demonstrate that the humanization event alone does not result in an ocular phenotype, and this is to ensure that our He9 mouse will serve as a robust control during CRISPR therapy development. In vivo, that is. And so if we were to expect to see an ocular phenotype, we'd expect to see features similar to those observed in the small eye or Say mouse. So we would expect to see cloudiness as well as vascularization of the cornea, and an eye that's smaller in size as well. And so here are some slit lamp images of a wild-type mouse as well as our He9 mouse bred on a B6 background, and we don't observe any ocular features that differ between these two images. I then went on to visually inspect 84 He9 mice as well as 90 wild-type mice and did not find a statistically significant difference between the observed phenotypes. This work was then repeated on a He9 mouse bred on a 129 background, and we found the same results. So this demonstrates that the minimal humanization event does not result in an ocular phenotype in mouse. So once we validated this model, I then went on to test various CRISPR strategies to correct the patient variant that we had just introduced into our mouse embryonic stem cells. And so some of this work is summarized here. Here we have a reference chromatogram of our unedited homozygous mutant cell line, and I'll direct your attention to the patient variant here. So we see one dominant red peak indicating 100% homozygosity for the patient variant. And when we introduce the first set of conditions with SP-Cas9 into the cells at the site of the patient variant, we can see that the base has now been called as a cytosine, and a high degree of correction is indicated by this dominant blue peak here. When we introduce the second set of conditions into the cells, you can already tell right off the bat, looking at the site of the patient variant, that this was not as successful in correcting the variant as indicated by the thymine base call. And finally, the introduction of the base editor system proved to show a very high degree correction of the patient variant. And I'll also note how clean you can tell the sequence is. So in the other conditions, we can see that secondary sequence popping up there indicating the presence of indels, and we don't see that through the base editor here. So these results were quantified, and we found that the base editor had the highest average correction at 75%. And when we introduced these conditions into the non-mutant cell line, we found that the base editor had the lowest impact on non-mutant chromosomes, which is ideal for the purpose of therapy development. And with that, I'd like to thank you for your attention, and I'm happy to take questions afterwards. Thank you.
Thank you, Bethany.